Hi everyone, welcome to the PowerPoint and video lecture for Module 10.2.2. Here we're going to discuss the higher order functions and continuation of the previous module. So higher order functions share three characteristics. One, the cerebral cortex is required for their performance, and they involve complex interactions among the areas of the cortex between the cerebral cortex and other areas of the brain. Two, they involve both conscious and unconscious information processing. Three, they are not part of the program wiring of the brain, and for this reason, higher order functions are subject to adjustment over time. In this section, we'll consider the mechanisms of memory and learning, and we'll also describe the neural interactions responsible for consciousness, sleep, and arousal. So what was the topic of the last sentence that I said? What's your social security number? What does a hot dog taste like? To answer these questions, you have to access memories, stored bits of information gathered through experience. Fact memories are specific bits of information, such as the color of a stop sign or the smell of perfume. Skill memories are learned motor behaviors. You can probably remember how to light a match or open a screw-top jar, for example. With repetition, skill memories become incorporated at the unconscious level. Examples include the complex motor patterns involved in skiing, playing the violin, and similar activities. Skill memories related to program behaviors, such as eating, are stored in appropriate portions of the brainstem. Complex skill memories involve the integration of motor patterns in the basal nuclei, the cerebral cortex, and the cerebellum. Two classes of memories are recognized. Short-term memories do not last long, but while they persist, the information can be recalled immediately. Short-term memories contain small bits of information, such as a person's name or a telephone number. Repeating a phone number or other bits of information reinforces the original short-term memory and helps to make sure that it gets converted to a long-term memory. Long-term memories last much longer, in some cases for an entire lifetime. The conversion from short-term to long-term memory is called memory consolidation, and there are two types of long-term memory. Secondary memories are long-term memories that fade with time and may require considerable effort to recall. And tertiary memories are long-term memories that are with you for a lifetime, such as your name or the contours of your own body. Memory consolidation and access. The amygdaloid body, or the amygdala, and the hippocampus, two components of the limbic system, are essential to memory consolidation. Damage to the hippocampus leads to an inability to convert short-term memories to new long-term memories, although existing long-term memories remain intact and accessible. Tracks leading from the amygdaloid body to the hypothalamus may link memories to a specific emotion. The nucleus basalis, or cerebral nucleus near the diencephalon, plays an uncertain role in memory storage and retrieval. Tracks connect this nucleus with the hippocampus, the amygdaloid body, and all areas of the cerebral cortex. Damage to this nucleus is associated with changes in emotional states, memory, and intellectual function. And we'll see that as we discuss Alzheimer's disease. Most long-term memories are stored in the cerebral cortex. Conscious motor and sensory motor memories are referred to in the appropriate association areas. For example, visual memories are stored in the visual association area, and memories of voluntary motor activity are stored in the premotor cortex. Special portions of the occipital and temporal lobes are crucial to the memories of faces, voices, and words. In at least some cases, a specific memory probably depends on the activity of a single neuron. For example, in one portion of the temporal lobe, an individual neuron responds to the sound of one word and ignores others. A specific neuron may also be activated by the proper combination of sensory stimuli associated with a particular individual, such as your grandmother. A result of these neurons are called grandmother cells. Information on one subject is parceled out to many different regions of the brain. And your memories of cows are stored in the visual association area, like what a cow looks like, that the letters C-O-W mean cow. The auditory association area, or the MU, sound and how the word cow sounds. The speech center, how to say the word cow, and the frontal lobes, how big cows are and what they eat. Related information, such as how you feel about cows and what milk tastes like, is stored in other locations. If one of these storage areas is damaged, your memory will be incomplete in some ways. How these memories are accessed and assembled on demand remains still a mystery. Memory consolidation at the cellular level involves anatomical and physiological changes in the neurons and synapses. For legal, ethical, and practical reasons, scientists do not conduct much research on these mechanisms with human subjects. Research on other animals, commonly those with relatively simple nervous systems, has indicated the following mechanisms may be involved. Increased neurotransmitter release. So, a synapse that is frequently activated increases the amount of neurotransmitter it stores and releases more with each stimulation. The more neurotransmitter release, the greater effect on the postsynaptic neuron. The facilitation of synapses. When a neural circuit is repeatedly activated, the axon terminals begin continuously releasing neurotransmitter in small quantities. The neurotransmitter binds to receptors on the postsynaptic membrane, producing a graded depolarization that brings the membrane closer to threshold. The facilitation that results affects all neurons in the circuit. The formation of additional synaptic connections. Evidence indicates that when one neuron repeatedly communicates with another, the axon tip branches and forms additional synapses on the postsynaptic neuron. As a result, the presynaptic neuron has a greater effect on the membrane potential of the postsynaptic neuron. And these processes create an anatomical change that facilitate communication along a specific neural circuit. This facilitated communication is thought to be the basis of memory storage. A single circuit that corresponds to a single memory has been called a memory engram. This definition is based on function rather than structure, and we know too little about the organization and storage of memories to be able to describe neural circuits involved. Memory engrams form as a result of experience and repetition. Repetition is crucial. That's why you probably need to read all of our chapters a little bit more than once before you take an exam. Efficient conversion of a short-term memory into a memory engram takes time, usually at least an hour. Whether that conversion will occur depends on several factors. They include the nature, intensity, and frequency of the original stimulus. Very strong, repeated, and exceedingly pleasant or unpleasant events are most likely to be converted to long-term memories. Drugs that stimulate the CNS, such as caffeine and nicotine, may enhance memory consolidation through facilitation. The hippocampus plays a key role in consolidating memories. The mechanism remains unknown, but it's linked to the presence of NMDA, or N-methyl D-aspartate, receptors, which are chemically gated calcium channels. When activated by the neurotransmitter glutamate, the gates open and calcium enters the cell. Blocking NMDA receptors in the hippocampus prevents long-term memory formation. So a quick note. Amnesia is the loss of memory as a result of trauma or disease. The type of memory loss depends on the specific regions of the brain affected. For example, damage to the auditory association areas can make it difficult to remember sounds. Recovery may be partial or complete. In retrograde amnesia, the person loses memories of events that occurred before the trauma or the onset of the disease. Some degree of retrograde amnesia commonly follows head injury. After a fall or a car wreck, many victims are unable to remember the moments preceding the accident. In anterograde amnesia, the person is unable to store additional memories, but earlier memories remain intact and accessible. The problem appears to involve an inability to generate long-term memories. Some degree of anterograde amnesia is common as a sign of senility. At least two drugs, diazepam or Valium and triazolam or Halcon, have been known to cause brief periods of anterograde amnesia. Brain injuries can cause more prolonged memory problems. A person with permanent anterograde amnesia lives in surrounding areas that they are always knew. The person can read magazines, chuckle over them, and reread them a few minutes later with equal pleasure. Clinicians must introduce themselves at every meeting, even if they have been treating this patient for years. When I talk about permanent anterograde amnesia, the movie 50 First Dates comes to mind. In post-traumatic amnesia, or PTA, a head injury produces a combination of retrograde and anterograde amnesias. The individual can neither remember the past nor consolidate memories of the present, and the extent and duration of the amnesia varies with the severity of the injury. The difference between a conscious individual and an unconscious individual might seem obvious. A conscious individual is alert and attentive, and an unconscious individual is not. Yet there are many degrees in both states. Conscious implies an awareness and attention to an external event and stimuli, but a healthy conscious person can be nearly asleep, wide awake, or high-strung and jumpy. Unconscious can refer to conditions ranging from deep, unresponsive states induced by anesthesia before major surgery to deep sleep to the light drifting nod that occasionally plagues students who are reading anatomy and physiology textbooks. A person's degree of wakefulness at any moment indicates the level of ongoing CNS activity. When you're asleep, you're unconscious but can still be awakened by normal sensory stimuli. Healthy individuals cycle between the alert, conscious states and sleep each day. When CNS function becomes abnormal or depressed, the state of wakefulness can be affected. An individual in a coma, for example, is unconscious and cannot be awakened even by a strong stimuli. So we recognize two general levels of sleep, each with characteristic patterns of brainwave activity. In deep sleep, also called slow wave or non-REM, your entire body relaxes and the activity of the cerebral cortex is at a minimum. Heart rate and blood pressure, respiratory rate, and energy use decline by up to about 30%. And during rapid eye movement or REM sleep, you dream actively and your blood pressure and respiratory rate change. Although the EEG that you see here resembles that of the awake state, you become even less receptive to outside stimuli than in deep sleep, and your muscle tone decreases markedly. Intense inhibition of somatic motor neurons probably prevents you from acting out the responses you envision while dreaming. The neurons controlling your eye muscles escape this inhibitory influence, and your eyes move rapidly as you dream, and that continues to unfold. Periods of REM and deep sleep alternate throughout the night, beginning with the deep sleep for about an hour and a half, and rapid eye movements periods initially average about 5 minutes, but over an 8-hour night, they gradually increase to about 20 minutes. Each night, we probably spend less than 2 hours dreaming, but variation among individuals is significant. For example, children spend more time in REM sleep than adults do, and extremely tired individuals have very short and infrequent REM periods. Sleep produces only minor changes in physiological activities of other organs and systems, and none of these changes appear to be essential to normal function. The significance of sleep must lie in its impact on the CNS, but the physiological or biochemical basis remains to be determined. We do know that protein synthesis in neurons increases during sleep. Extended periods without sleep lead to a variety of disturbances in mental function. Roughly 25% of the U.S. population experiences some form of sleep disorder. Examples include abnormal patterns or durations of REM sleep or unusual behaviors during sleep, such as sleepwalking. In some cases, these problems affect the individual's conscious activities. Slowed reaction times, irritability, and behavioral changes may be a result. Memory loss has also been linked to sleep disorders. As we get into some more disorders, I thought it'd be a good idea to share that neurotissue is extremely delicate, and the characteristics of its extracellular environment must be kept within a narrow homeostatic limit. Signs and symptoms of neurological disorders appear when homeostatic regulatory mechanisms break down, and this may happen under the stress of genetic or environmental factors such as infection or trauma. Literally hundreds of disorders affect the nervous system, and they can be categorized as infections, which include diseases such as rabies, meningitis, and polio, congenital disorders such as spinal bifida and hydrocephalus, degenerative disorders such as Parkinson's disease and Alzheimer's disease, tumors of neural origin, trauma such as spinal cord injuries and concussions, toxins such as heavy metals and the neurotoxins found in certain seafoods, secondary disorders, which are problems resulting from dysfunction in other systems, for example, strokes and several demyelination disorders. And a standard physical examination includes a neurological component. The physician checks the general status of the CNS and the PNS. In neurological examinations, a physician attempts to trace the location of a specific problem by evaluating the sensory, motor, behavioral, and cognitive functions of the nervous system. Arousal, or awakening from sleep, appears to be one of the functions of the reticular formation. The reticular formation is especially well suited for providing watchdog services because it has extensive interconnections with the sensory, motor, and integrative nuclei and pathways along the brainstem. Your state of consciousness results from a complex interaction between the reticular formation and the cerebral cortex. One of the most important brainstem components is a diffuse network in the reticular formation known as the reticular activating system, or RAS. This network extends from the medulla oblongata to the midbrain. The output of the RAS projects to the thalamic nuclei that influence large areas of the cerebral cortex. When the RAS is inactive, so is the cerebral cortex, and the stimulation of the RAS produces a widespread activation of the cerebral cortex. The midbrain portion of the RAS appears to be the headquarters of the system. Stimulating this area produces the most pronounced and long-lasting effects on the cerebral cortex. Stimulating other portions of the RAS seems to have an effect only to the degree that it changes activity of the midbrain region. The greater the stimulation of the midbrain region of the RAS, the more alert and attentive the individual will be to incoming sensory information. The thalamic nuclei associated with the RAS may also play an important role in focusing attention on specific mental processes. Sleep may be ended by any stimulus sufficient enough to activate the reticular formation and RAS. Arousal occurs rapidly, but the effects of a single stimulation of the RAS last less than a minute. After that, consciousness can be maintained by positive feedback, because activity of the cerebral cortex, the basal nuclei, and the sensory motor pathways will continue to stimulate the RAS. After many hours of activity, the reticular formation becomes less responsive to stimulation. You become less alert and more lethargic. The precise mechanism remains unknown, but neural fatigue probably plays a relatively minor role in the decreasing RAS activity. Evidence suggests that the regulation of sleep-wake cycles involves an interplay between brainstem nuclei that use different neurotransmitters. One group of nuclei stimulates the RAS with norepinephrine and maintains the awake alert state. Another group depresses the RAS activity with serotonin, promoting deep sleep. These dueling nuclei are located in the brainstem. Changes in the normal balance between two or more neurotransmitters can profoundly affect the brain. Recall from previous sections that the interplay between populations of neurons releasing norepinephrine and serotonin appears to be involved in regulating sleep-wake cycles. Another example concerns Huntington's disease. The primary problem in this inherited disease is the destruction of ACH secreting and GABA secreting neurons in the basal nuclei. The reason for this destruction is unknown. Symptoms appear as the basal nuclei and frontal lobes slowly degenerate. An individual with Huntington's disease has difficulty controlling movements and intellectual abilities that gradually decline. In many cases, the importance of specific neurotransmitter was revealed while searching for the mechanism and activation of other drugs. These examples are as follows. It's now clear that an extensive network of tracts delivers serotonin to nuclei and higher centers throughout the brain, and various or variations in serotonin levels affect sensory interpretation and emotional states. Compounds that affect serotonin produce hallucinations. For example, lysergic acid diethylamine, or LSD, is a powerful hallucinogenic drug that activates serotonin receptors in the brainstem, hypothalamus, and limbic system. Compounds that merely enhance the effects of serotonin also produce hallucinations, where compounds that inhibit serotonin produce or block this activation and cause severe depression and anxiety. An effective antidepressive drug, now in widespread use, is all over the place, is called fluoxetine, or by its brand name Prozac. And that slows the removal of serotonin at synapses, causing an increase in serotonin concentrations at the postsynaptic membrane. Such drugs are classified as selective serotonin reuptake inhibited, or SSRIs. Other important SSRIs include Celexa, Luvox, Paxil, and Zoloft. Norepinephrine is another important neurotransmitter with pathways throughout the brain. Drugs that stimulate norepinephrine release cause exhilaration, and those that depress norepinephrine release cause depression. One inherited form of depression has been linked to defective enzyme involved in NE synthesis. Disturbances in dopamine transmission have also been linked to several neurological disorders. We've already seen that an inadequate dopamine causes motor problems in Parkinson's disease. An excessive production of dopamine may be associated with schizophrenia, which is a psychological disorder marked by pronounced disturbances of mood, thought patterns, and behavior. Amphetamines, or speed, stimulate dopamine secretion, and in large doses can produce symptoms resembling those of schizophrenia. Dopamine is thus important not only to the nuclei involved in the control and intentional movements, but in other centers of the diencephalon and cerebrum. As a quick note, Alzheimer's disease is a progressive disorder characterized by the loss of higher order cerebral functions. It is the most common cause of senility. Symptoms appear around age 50 or 60 and later, although the disease occasionally affects younger people as well. Alzheimer's disease has widespread impact. An estimated 5.2 million people in the United States, including about 11% of those over the age of 65 and 38% over the age of 85, have some form of the condition. It causes approximately 100,000 deaths a year. The link is uncertain, but microscopic examination of the brain shows abnormal plaques and neurofibrillary tangles in the regions involved with memory, emotion, and intellectual function. It's still unknown whether these deposits cause Alzheimer's disease or are secondary signs of ongoing metabolic alterations with an environmental, hereditary, or infectious basis. Genetic factors certainly play a minor role. The late onset form of Alzheimer's disease has been traced to a gene on chromosome 19 that codes for proteins involved in cholesterol transport. Fewer than 5% of people with Alzheimer's disease have the early onset form, which may develop before age 50. And this form of Alzheimer's disease has been linked to genes on chromosomes 1, 14, 19, and 21. Interestingly, the majority of individuals with Down syndrome develop Alzheimer's disease relatively early on in life. And for those of you who don't know, Down syndrome results from an extra copy of chromosome 21, and it's also known as trisomy 21. So there's no cure for Alzheimer's disease. A few medications and supplements slow its progress in many patients. Diagnosis involves excluding metabolic disorders, chemical and drug interactions, nutritional deficiencies, and anatomical conditions that can mimic dementia. Taking a detailed history and doing a thorough physical exam and evaluating mental function and mood. Depression may also mimic dementia in some cases. The aging process affects all body systems, and the nervous system is no exception. Anatomical and physiological changes probably begin around the age 30 and accumulate over time. An estimated 85% of people above the age of 65 lead relatively normal lives, but they exhibit noticeable changes in mental performance and in CNS function. Common age-related anatomical changes in the nervous system may include the following: reduction in brain size and weight. This reduction results primarily from a decrease in the volume of the cerebral cortex. The brain of elderly individuals has a narrow gyre and wider sulci than those of young people, and the subarachnoid space is larger. Also includes a reduction in the number of neurons. Brain shrinkage has been linked to a loss of cortical neurons, although evidence indicates that neurons are not lost, at least to the same degree, in brain stimuli nuclei. It also includes a decrease in blood flow to the brain. With age, fatty deposits gradually build up in the walls of blood vessels. Just as a clog in a drain reduces water flow, these deposits reduce the rate of blood flow through the arteries. This process is called arteriosclerosis and affects arteries throughout the body. Even if the reduction in blood flow is not large enough to damage neuron, arteriosclerosis increases the chance that the affected vessel wall will rupture, damaging the surrounding neurotissue and producing signs and symptoms of a cerebrovascular accident, or CVA, also called a stroke. In many areas, the number of dendritic branches, spines, and interconnections appears to decrease. Synaptic connections are lost, and the rate of neurotransmitter production declines. Many neurons in the brains accumulate abnormal intracellular deposits, including lipofuscin and neurofibrillary tangles. Lipofuscin is a granular pigment with no known functionality. Neurofibrillary tangles are masses of neurofibrils that form dense mats inside cell bodies and axons. Plaques are extracellular accumulations of fibrillar proteins, surrounded by an abnormal dendrites and axons. Both plaques and tangles contain deposits of several peptides, primarily two forms of amyloid beta protein, the fibrillar and the soluble. They also appear in brain regions such as the hippocampus, specifically associated with memory processing. Their significance is relatively unknown. Evidence indicates, though, that they appear in all aging brains. In excess, they seem to be associated with clinical abnormalities. The anatomical changes associated with aging of the nervous system affect all neural functions. For example, memory consolidation typically becomes more difficult, and secondary memories, especially those of recent past, become harder to access. The sensory systems of elderly people, notably hearing, balance, vision, smell, and taste, become less sensitive. Lights must be brighter, sounds have to be louder, smells have to be stronger before they are perceived in the elderly. Reaction rates are slowed, and reflexes, even some withdrawal reflexes, weaken or disappear. The precision of motor control decreases, and it takes longer for an elderly person to perform a given motor pattern than it did 20 years earlier. For roughly 85% of the elderly population, these changes do not interfere with their abilities to function in society. But for reasons yet unknown, some elderly individuals become incapacitated by progressive CNS changes. These degenerative changes, which can include memory loss, anterograde amnesia, and emotional disturbances, are often lumped together as senile dementia or senility. By far the most common and disabling form of senile dementia is Alzheimer's disease.